Welcome to our video where we will cover some of the more important morphological characters used to identify thrips and include a video walkthrough demonstration showing how to use a Lucid key. My name is Ben Diehl. I'm an entomologist at Washington State University. Today I'll be discussing some of the research I did as part of the task grant with the goal of assisting those interested in identifying thrips that were found on peonies grown in Alaska. First, let's start by including some details on thrips taxonomy. Thrips belong to the order Thysanoptera, which is divided into two main suborders, Terebrantia and Tubulifera. Both groups were recovered from Alaska peonies, so it's important to recognize the difference. The suborder Terebrantia includes the majority of thrip species that were collected during our task research and can be recognized by the cone-shaped final segment of female specimens. Females also have a saw-like ovipositor that is used to insert eggs into plant tissues. The Tubulifera were uncommonly collected in peony samples and have the last abdominal segment tube-shaped in adults. Species belonging to this group also lack an ovipositor and instead lay eggs directly onto the surface of plants. Next, let's cover how to tell the difference between adult male and female thrips. In general, male thrips were uncommonly represented in samples from peonies, with the exception of the species Thrips orionis, where males made up 64% of specimens recovered. Males are often smaller than females and tend to have narrower abdomens, as you can see from the comparison photos on the right. Many species of male thrips also have structures on their sternites called pore plates. The shape of these can be used to differentiate species. Lastly, some males possess finger-like terminalia at the tip of their abdomen. These structures may look similar to the ovipositors of adult females, but lack the saw-like teeth. We will start our tour of body characters on the head. The antennae hold a number of useful structures used in identification, but we will focus on the number of segments. Counting the number of antennal segments can be a good way to quickly narrow down the list of possible species, especially when using the Lucid key. It is important to know that some species may have a variable number of antennal segments, for example, usually having 7, but sometimes 8, due to natural variation. Moving to the top of the head, we will look next at the ocelli. These sensory structures are capable of detecting changes in light and movement, but not in the visual detail made possible by the compound eyes. Most thrip species possess 3 ocelli arranged in a configuration referred to as the ocellar triangle. A single species of thrips found on peonies during this project, Thrips secticornis, not only lacks ocelli, but also wings. Interestingly, these two characters appear to be linked and are often absent together in insects. Situated around the ocelli and between the compound eyes are several pairs of ocellar setae, or hair-like structures. The number and position of these pairs of setae are diagnostic. Commonly, there are 3 pairs of ocellar setae, called pair 1, pair 2, and pair 3. But some species have more. All species of thrips in the genus Thrips lack pair 1. The position of pair 3 in relation to the ocellar triangle is important, as well as the length of the setae in comparison to the sides of the ocellar triangle. Moving to the thorax, we will explore 2 characters located on the metanotum. The first is the position of the median setae, indicated by a blue line, in relation to the anterior margin, indicated by a red line, of the metanotum. There are 3 different states for the position of the pair of median setae: arising at the anterior margin, as in figure A, arising somewhat behind the anterior margin, as seen in figure B, or arising near the posterior margin, indicated by a yellow line, as in figure C. Also located on the metanotum are the Campaniform Syncilla. These are sensory organs that lack an external process and resemble a circular pore in the surface of the exoskeleton. They are typically located near the posterior margin, indicated by a yellow line, as before, of the metanotum. There are 2 main states to this character. A pair of syncilla are present. In some cases, only a single syncillum is present, or the Campaniform Syncilla are absent. In some species, like the western flower thrips, Franklinelia occidentalis, this character is somewhat plastic and can be present, absent, or have only one syncillum of the pair. Finally, we move to the abdomen, more specifically to the tergites, or the segments located on the dorsal surface of the abdomen. One of the most important morphological characters for separating thrips collected from Alaska-grown peonies are the paired Tinidia that are located on the lateral areas of tergites 5 through 8, or sometimes fewer. These are comb-like structures found on the tergites of two commonly encountered genera, Frankliniella and Thrips. In order to tell these two taxa apart, the position of the Tinidia, indicated by blue line, in relation to the spherical, indicated by a red letter S, is noted. In Frankliniella species, the Tinidia are anterolateral to the spherical, while in Thrips species, the Tinidia are posteromesad to the spherical. The final character we will discuss is also on tergite number 8. The posteromarginal comb is comprised of a line of microtrichia and is located on the posterior edge of tergite 8. There are 3 main character states. The comb runs completely across the posterior margin, as seen in figure A. The comb is incomplete medially, as in figure B, or the comb is absent entirely, as seen in figure C. The shape and length of the microtrichia making up the posteromarginal comb are also used to separate some Thrips taxa. Now that we have talked about some of the most important morphological characters used to identify Thrips collected in Alaskan peonies, let's take a brief look at how the Lucid Key is different than a traditional dichotomous one, and the process of using a Lucid Key to identify an unknown Thrips specimen. Lucid Keys allow a person more freedom to arrive at an identification because the user is not bound to a predetermined path of sequential characters selected by the author of the key. In a dichotomous key, if a user becomes stuck on a certain couplet due to confusion about character states or a specimen that is lacking body parts, it is difficult to progress further. In Lucid Keys, however, the user can pick and choose which characters to select in any order. Because Lucid Keys are deployed in an interactive digital format, they also allow for the inclusion of more photographs or other resources that can aid in species identification. Let's take a look at an example of how to use a Lucid Key that was developed for the task project. Here is the user interface of the Lucid Key. You can see there are 4 windows containing information on the characters and species that are included in the key. The top left window allows you to browse and select the characters you want to score. Characters you select are then placed into the bottom left window, showing a history of which characters you used to arrive at an identification. The top right window shows all of the taxa that are remaining as possibilities for the identity of your unknown Thrips. Once you start selecting and scoring characters, species that are excluded from the key will move down to the bottom right window. If you change your mind about a character and either rescore or unselect the character altogether, any changes in which species are now included or excluded will also be represented in these 2 windows in real time. First, let's start on the head and look at the Ocelli. If you would like to view photographs that show examples of each character state, you can click on the thumbnail icon next to each of the choices. Additionally, you may click on the Feature Thumbnails button in the toolbar to open all photographs at once. Our unknown specimen does have Ocelli, so we click Present. We can see on the bottom right window that Apterothrips secticornis was excluded as a possible identity because it is the only species in the key that lacks Ocelli. Let's pretend that our specimen is damaged and the antennae are missing. For this reason, we will not score the number of antennal segments. We will move to the metanotum and score the median CD as arising at the anterior margin, and score the Campaniform Syncilla as present, a pair of Syncilla. Moving to tergite 8 on the abdomen, we will score the paired Tinidia as present, anterolateral to the spherical, and the posteromarginal comb as complete. With this final character scoring, you can see that we are left with only 1 species in the top right window. Frankliniella occidentalis, commonly known as the Western flower thrips. If you would like to view a zoomed out photograph of a Frankliniella occidentalis adult to see if it matches your recently identified specimen, you can click the thumbnail icon next to the species name. For some species, a photograph of both males and females are available. Because it is always possible that unsampled or newly introduced thrips could show up on peonies grown in Alaska in the future, it is always a good idea to double check identifications if your unknown specimen does not exactly match one of the species included in the key by scoring any remaining characters in the Lucid key or using the dichotomous key that was also developed as part of the task grant. Thank you for watching this video and best of luck on your identifications.